IR region present on the right IR region is the T3, C, A, T, C3 and TA. It is this is the right inverted repeat region. This is the left inverted repeat region. Presence of a ORF, open reading frame region, and this is the main segment, main part of this. You know that the transposon has the transposase encoding gene. This segment I have written here. This is the transposase encoding gene which encodes the transposase, and this AC element which is a transposon of the MAGE consists of look here, consists of how many base pair? Consists of I have written here. This consists of 4.56 kbp. The length of this DNA, length of this transposon is 4.56 kbp, or you can also write 4563 base pair लिख सकते हो. You can write this AC element of the MAGE consists of 4563 base pair, or you can also write in this way 4.56 kbp. किलो बेस पेयर में लिखे या फिर आप बेस पेयर में लिखे. So the length of this DNA, the length of this AC element of the MAGE is 4.5 kbp, and this transposase encoding gene of this activator element is transcribed to produce mRNA, and the length of the mRNA is 3.5 kbp, and the molecular weight of the mRNA is 93 kDL. Okay? So this is the structure of the AC element activator element of the MAGE. Now look at the dissociator element. This is the second diagram I have shown here. The DS element of the MAGE. DS stands for dissociator element. This dissociator element is the truncated part of the AC element, from which the AC element gives rise to the formation of dissociator element. When some part of the DNA of some segment of the AC element undergoes deletion, and deletion results in the production of a dissociator element, and this dissociator element is of three types: dissociator element A, dissociator element B, and dissociator element C. What is the difference between dissociator element A, dissociator element B, and dissociator element C? Look, if a small part of the AC element undergoes deletion, then it results in the production of dissociator element A. When a larger part of the segment of the activator element undergoes deletion, it leads to the production of the second type of this dissociator element. And what happens in case of the third type of the dissociator element? Dissociator elements are of the three types, as you have seen on the whiteboard: dissociator element A, dissociator element B, and dissociator element C. In case of dissociator element C, a larger portion of the basic transposon undergoes deletion, and you look. But in all the cases, the inverted repeat region remains intact. Inverted repeat regions are not lost. The inverted repeat region, right inverted repeat region, left inverted repeat region, and this is the part of the transposon. So inverted regions are not deleted. Rather, the inverted regions of the remains intact. Only the central part, that is the part of the DNA constituting the transposons. Main part of the transposons is deleted. Some part or a larger part of the transposing area is deleted. What does it show? It shows that the AC element, that is the activator element, is the main transposon, whereas the dissociator element is the truncated part of the activator element, and it is formed due to deletion of a part of the DNA from the activator region. Now look at the features of these elements. Hello.
Yes, sir.
Now let us discuss some of the important feature of the activator element and some of the important feature of the dissociator element. Activator the first basic, the first main point of this activator region is that this activator region is responsible for the coloration. In the previous lecture, you have seen I had discussed that the kernel, the maize grain may have some colored grains, and the grain may be purple, the grain may be brown, or yes. So the region, this activator region, this activator segment or the activator transposons are responsible for the development of the coloration in the maize grain. And how does this coloration take place? This activator region brings about the deletion of the knob. You know that knob is present at the end of the 9th chromosome of the maize, and just before the knob, the genes, the very important gene, I have written that the inhibitory gene, CI genes and BZ gene. These two genes are very important, which are responsible for, and the CI gene, that is the inhibitor gene present on the 9th chromosome of the maize, is responsible for inhibiting the color development. The maize you have seen in the market are not brown, are not purple. Rather, mostly you see the normal maize grain,
Yeah.
and this normal phenotype is due to the presence of the inhibitory CI gene, which I have already discussed in the previous lecture. CI gene inhibits the expression of the gene responsible for the development of the color. And how does this inhibitory genes get lost? This activator region, this activator element, which is the transposon of the maize, this is the transposon which is responsible for the controlling or regulating the color development in the maize grain. And how does this coloration take place? Because this activator region of the activator element, which is the transposon of the maize present on the 9th short arm of the 9th chromosome, it brings about the deletion, removal of the CI gene, inhibitory gene, and BZ gene, so that the color grains may be formed on the cob. So this transposition, the transposon which is known as the activator element of the maize, which is also present on the 9th chromosome of the maize, and the genes and the inhibitory genes which are responsible for inhibiting the color development are also available on the chromosome. And when this transposon AC element brings about the deletion of the CI inhibitory gene, when the transposition takes place, what happens? When the transposition takes place, the CI gene, the inhibitory gene, and the BZ gene, capital B, capital Z gene. I think that you do not have in your memory the CI genes and the BZ gene. I think that, have you gone through the notes provided in the previous lecture, in the introductory lecture on this is the third lecture, Ujma. This is the third lecture on the eukaryotic transposon. First lecture during my first lecture on the transposon, I have described all the terms, BZ gene, CI genes. So go through the notes provided during the first lecture. It will become easier to understand everything. So this is the activator transposon of the maize, which brings about the development of the coloration of the maize grain by deleting the CI genes and the BZ gene. And if these two genes which inhibit the phenotypic expression of the aileron coloration, if they are lost, what will happen? The colored aileron will be formed. Therefore, this activator transposon of the maize, which is known as the SC element of the maize, this is very important. And in the second lecture on transposon, I have also made it clear that the transposition, if a particular transposon gets translocated, gets inserted at a particular site, what it does? it brings about either activation or inactivation of the adjacent gene. पिछले लेक्चर में मैंने आपको बताया है कि ट्रांसपोजोन का ये कैरेक्टर होता है। क्या कैरेक्टर होता है? कि वह जहां जाता है टारगेट साइट पर जहां इंजर्ट होता है, उसके बगल वाले जीन को या तो एक्टिवेट कर देगा या उस जीन के एक्सप्रेशन को इनएक्टिवेट करता है। दोनों काम कर सकता है। ये किसी ट्रांसपोजिशन के बाद ये जो ट्रांसपोजिशन का जो टारगेट साइट होता है, जहां से ट्रांसपोजोन उठकर जाएगा जहां अटैच होने के लिए, वहां पर के अगल बगल का जो जीन होता है, जो नियर बाय जीन्स होते हैं, उनको या तो एक्टिवेट कर देंगे या उनको क्या कर देंगे, उनके एक्सप्रेशन को इनएक्टिवेट कर देंगे। अब ये जो ट्रांसपोजोन जो आप देख रहे हैं, कौन सा? एसी एलिमेंट, जिसको एक्टिवेटर एलिमेंट जो कह रहे हैं, जो मेज का फर्स्ट ट्रांसपोजोन रिपोर्टेड है। मेज का ही क्या, पूरे दुनिया में जो पहला ट्रांसपोजोन डिस्कवर हुआ यही है। तो ये इनका जब ट्रांसपोजिशन होता है तो ये क्या करते हैं, जो एडजेसेंट जीन है, कौन वाले जीन, जो सीआई जीन है, जो इनहिबिटरी जीन है, जो बीजेड जीन है, उनको क्या करता है, वहां से उनको डीएक्टिवेट कर देता है, क्या कर देता है, इनएक्टिवेट करता है। कैसे? बाय ब्रिंगिंग अबाउट द डिलीशन ऑफ दो जीन। अलग अलग मेथड्स है इनएक्टिवेशन का। यहाँ क्या करता है ये, कि ये जो एक्टिवेटर एलिमेंट जो ट्रांसपोजोन है, जो अपने टारगेट साइट पर जब ये जाएगा तो वहां क्या कर देगा, जो एक्टिवेटर रीजन, जो कौन जीन है जी, सीआई जीन जो है, जो कलर फॉर्मेशन को रोकता है, और जो बीजेड जीन है, उन दोनों जीन को वहां से डिलीट कर देगा। तो बताओ कि जो कलरेशन को इनहिबिट करने वाला जीन है, जब वही वहां से इफ द जीन्स आर लॉस्ट, व्हाट विल हैपन? The coloration of the aileron layer will take place. Therefore, this transposon, the activator element of the mage, which is present on the short arm of the 9th chromosome of the mage, plays a very important role in the phenotypic in the regulation of phenotypic expression of the color in the aileron layer of the mage grain. So in this way, this activator element brings about the regulation of the expression of coloration in the aileron by removing the inhibitory genes and BZ gene. Now look at, this is its very important character. Now look at the dissociator element. Dissociator element, you know that dissociator element is not a new transposon. Rather, it is the product of deletion. Product of deletion of the activator region. When deletion takes place, when some part of the DNA from the activator region is deleted, it gives rise to the production of dissociator region. And this dissociator elements are of three types: DSA element, DSB element, and DSC element. And these three types of dissociator element depends upon the size of the segment of the DNA deleted from the activator region. If so in the first case, larger part of the DNA is removed. In the second case, a smaller part, and in the dissociator C, the very small part of the DNA remains. And in case of DSA, what happens? In case of dissociator A, a small part of the DNA is removed. In case of dissociator B, a large part is removed, and in case of third type, a very large part is deleted. But the inverted repeat regions remains tagged as it is. जो इनका जो inverted repeat region होता है,